We're going to end up looking at one last feature that is associated with the polarity of water molecules and then the property of water. And we've seen from these different things that water can form hydrogen bonds that allow different water molecules to sort of attach weakly to each other. Think of them like really small, weak magnets. Individually, they're easy to break. But if you line up a massive amount of hydrogen bonds, that's going to make it harder and harder to break things apart, which is why water can follow or can form things like ice as it changes matter, because you take out the heat energy that is causing those water molecules to spin, and that allows more and more hydrogen bonds to form, and eventually you freeze these water molecules in place. And remember, that makes ice less dense than liquid water, so ice floats on top, which is a great thing for life. But in this last thing, what we want to take a look at is how water can actually have this special property, because water, it turns out, can split into ions. So if we take our water molecule over here, and here we have a two-way arrow, which is what chemists use to refer to this equation, can go in both ways, and it will be in balance. So your solution of water can also have ions floating around in there. And over here we've got our proton. Remember, our proton is basically just that positively charged particle. So it's lost its electron because this has ionized right here, and now that electron is floating around this half of the water molecule. You've got what's known as a hydroxyl ion that has a negative charge associated with it. So when you've got water out there, it's also got other kinds of things floating around inside the H2O right there. And this is important because when we look at various water solutions that are out there, we can find that water actually is splitting apart, and we can measure the parts or the concentration of protons that are there. And so when you think of pH, think parts hydrogen. How much of this solution is actually in this form? And when chemists measure this, they found that it actually turned out to be a logarithmic function. And when you look at log functions here, a change in pH of 1 is a change in this thing that is 10 times. So if we look at a change over here for our pH scale from 6 to 7, there you're looking at a 10-fold difference in the concentration of your protons. If you're looking at a change from 5 to 7 over here, you've got 2 pH units, which means you're actually looking at 100 times the change in the number of those protons. And it turns out this is really important for cells because remember, 70-90% of a cell is water, and they're floating around in an aqueous environment typically. And when we look out there, we can measure the parts of hydrogen in a solution, and chemists refer to, if your pH is over here on this side of the scale, from 7 to 14, you're in what's known as an alkaline solution. And alkaline solutions tend to have a higher concentration of hydroxyl ions floating around in there. The opposite side of our pH scale is the acidic solution over here, and that's where you've got more protons floating around. So if you look at this, you can see more of the little pink protons, and your pH is on this side of the scale. So going from 7 all the way down to a pH of 0 around here. And we can also find right in the middle of the scale, when you look at distilled water or pure water, you're at what's known as a neutral pH. The amount of protons is balanced with the number of hydroxyl ions that are out there. And so you can see a variety of different solutions that are out there have different pHs. There's your stomach acids and your citrus fruits that are out there, and tomatoes. Those are all on the acidic side. And certain microbes can actually live in this pH range. There are some that can actually live down here. So yes, there are microbes that can indeed survive through your stomach acids that are out there. We've also got microbes that are over here in the alkaline side of this. These are things that we're going to tend to find in the soil that's out there. Certain kinds of extreme soil has got a very alkaline pH that's out there. You can feel this, and if you think about these extremes in pH on either side, we aren't adapted to either of these things. If you put your fingers in acids or an alkaline solution, you will actually feel your skin burning, especially the acid. You can see it, and the alkaline solution, your cells are basically going to explode. And if you rub your fingers together after putting them in a little bit of bleach or soapy water, you can actually feel that slipperiness, and that's actually your cells coming off. Most of the time, we're used to this neutral pH over here. But again, microbes can grow all over the place when we look at the pH there. So this is an important thing to remember when it comes to talking about growing cells in the lab. We definitely have to know what kind of pH they're used to and what kind of pH range they like to live in, because we want to be able to grow up things effectively in the lab. So we can actually control the parts of hydrogen in different kinds of solutions that are out there by adding different kinds of salts, and that can actually change the chemistry of a solution that's out there. If you add what's known as an acid, it will actually dissolve, and it's going to increase the amount of those protons that are floating around in the solution. And again, an easy way of looking at an acid, this is hydrochloric acid. Pretty easy. If you throw hydrochloric, well, you shouldn't throw it in. You should carefully pour hydrochloric acid into that water solution. Now you are going to see the hydrochloric acid is going to ionize, and you've just added a whole bunch of extra protons. So an acid is increasing the amount of those protons in the solution. A base, on the other hand, is the opposite, and a base is a chemical that when we add it, it's going to reduce the concentration of your protons in the solution there. And when you look at bases, they can basically function in two ways to reduce the amount of protons in your solution. This is sodium hydroxide, and when you take this, it'll come in little salt pellets, and you can just dissolve it into your water solution, and when this ionizes, it's going to release a sodium ion and your hydroxyl ion. And so if you think about that, I've donated more of these, which means I've just reduced the amount of protons there. And that's one way a chemical compound can act as a base. You can also have a second way over here, where the example I pulled out, a really simple one, is ammonia. There's ammonia. It's nitrogen. Remember, nitrogen, just like oxygen, is an electronegative element that's out there. And this one is going to coordinate with 3 hydrogens right there. Well, if you throw ammonia, again, carefully pour the ammonia into the water, it is actually going to bind up with this proton that's floating around there, and this is going to make an ammonium ion. Now you've got an ammonia solution, and you've just taken your protons out of the solution because now they're bound up with your ammonia. So both of these are going to be basic or alkaline compounds that are going to change the pH of a solution, because they're effectively reducing your number of proton ions that are floating around in there. And when we look at cells that are out there, hopefully one of the things you pick up from your other biology classes is that cells are in this constant state of change due to their environment. But we want to keep everything relatively the same inside. And that's where cells have a solution. They use chemicals that are known as buffers to resist changes in the environment due to pH changes. So if you have a microbe floating around in a river and it goes into an offshoot of the river where there's a whole bunch of an acidic compound dissolving in there, that cell's going to die. That change in pH is going to disrupt its cell processes. And cells are going to use chemicals out there to help to resist these rapid changes in pH. And so if we look at this idea, if you are in an environment where there's an excess amount of these protons, a buffer is going to suck up those protons. And then you leave that environment and you go to an environment where there's a lack of protons, that buffer can release the protons. So when we look at this, a buffer can act as a proton donor, or it can act as a proton acceptor. And I'll end off with this little guy, just to confuse you and piss you off a little bit. This is an example of a carbonate buffer. that we would find in your body, and this is floating around inside your bloodstream to help buffer the pH of your blood. And you have cells that are releasing carbon dioxide gas, and that gas is going to dissolve in the water. And when it does, it's going to form something known as carbonic acid that chemists would write out over here like this. This carbonic acid can actually donate protons to that solution. So one of those protons breaks off, and now you've got bicarbonate ion that is floating around in that solution. And bicarbonate ions can actually act as either a donor or it can act as an acceptor. And over here we end up, if it donates this other proton right there, and now you've got two of these protons that are floating around in your solution, now you've got this carbonate molecule right there. And that carbonate can accept one of these protons, and it can become bicarbonate that can then accept another proton and become carbonic acid, and then you can form water and you can release CO2. So this is going on inside of you right now when you're breathing and you're releasing this stuff from your cells. You need to get rid of the excess carbon dioxide. And so this is what you're hopefully going to be breathing out unless you've fallen asleep by now because of this chemistry introduction. So again, I'm trying to get people to think about the objectives. So what should you pick up from this chemistry stuff? Make sure that you can describe the structure of an atom. So think about the nucleus, protons and neutrons, and the orbiting electrons that are out there. Give me some examples of main elements that are making up most of the cell's mass, and hopefully you remember CHON, and think about the function for some of these elements and the different kinds of minor and trace elements that cells have out there. Make sure that you can describe the different types of bonds that atoms are going to use to form these molecules, and I gave you the two examples of covalent and also our ionic bonds. Likewise, be able to describe how covalent bonds can be polar and how this produces hydrogen bonds between the molecules that are out there. And the stuff I went into about hydrogen bonds, that's a really important thing when you look at life, because life evolved in water and it still has a bunch of water in it. So think about all these properties that are due to the hydrogen bonds and how that can give us hydrophobic and hydrophilic molecules that are either afraid or like water, amphipathic compounds that are out there that like and don't like. They are amphibians. They go in and out of water depending on the chemistry of it, whether or not that part of the molecule is polar or non-polar. And then the final thing is that idea of the parts of hydrogen, since water can ionize, and how this is going to have the effect of changing the chemistry of that solution when we can look at the amount or parts of hydrogen and how acids and bases are compounds that can change the pH of a solution and what buffers are used for.